Morning, sir.
Okay, can you see my PPT?
Not yet, sir.
Not
Okay, okay, just a second. Let me just make it. Now, is it visible?
Yes, sir.
Okay, so
yes.
let us start. In the previous class, we were talking about the basic concepts of metabolism. So you have learned that the molecules, they can be either synthesized or they can be broken down. When they are broken down, we are talking about the catabolism. When they are synthesized, we call them as an anabolism. And there is a very tight regulation by which they can produce a smaller molecule from macromolecule, or they can synthesize macromolecule from. And what you have learned is that all these processes which are occurring inside the cell, they are being controlled or they are being activated by enzymes. So we have also discussed about the concept of enzymes. You have learned that enzymes are the one which are regulating the reaction. It does not mean that enzyme starts the reaction or ends the reaction. It means that it helps in the process by which it can reduce the time when the equilibrium of the reaction can occur. So actually, if any reaction inside the cell is taking very long time because the reaction is extremely slow, what enzyme does is that it helps in attaining that equilibrium. That means reaction becomes faster, of course, both in forward direction as well as in backward direction, and that is why its equilibrium is attained much faster. Now, if I say that the enzymes are the one which are present for catalyzing all the reactions which are happening or occurring inside the cell, the question comes how the metabolism is regulated. Present, then how you can control a reaction, how we can say that the cell should make more macromolecule, our cell should produce more energy. So we are going to discuss that concept today. So here it is very clear that the metabolism is being regulated or controlled by three different processes. One of them could be that the amount of enzyme itself is not enough to catalyze the reaction or is absent. So what will happen if I control the amount of enzyme, then I can control the reaction. If I want to stop, for example, the catabolic reaction, I will, stop the production of catabolic enzyme, and that's why the catabolism will not happen. On other hand, if there is anabolism is to be stopped, we will stop the enzyme production from which is responsible for anabolism. So how do we do that? This is done by controlling the synthesis of enzyme itself. And how enzyme is being synthesized? Enzymes are proteins, so they will be synthesized in the same manner proteins are being synthesized. So they will be dependent on the rate of transcription process. So you can see that if I can change the rate of transcription where your DNA is forming mRNA, and this mRNA is going to synthesize a particular protein which acts as an enzyme, what you have done is that you have controlled the formation or production of that particular protein or enzyme, and that is why you can control the reaction. Is this clear to you?
Yes, sir.
Okay, let us move on. So first step is that I just stop the formation, or vice versa. We can also do in such a way that we can produce a lot of enzyme so that that particular process can occur. Now, this concept, I even experiment, the source, which is in the higher amount present outside the cell, then they will utilize the energy which is most easily utilizable than less easily utilizable. It's a very difficult concept to understand, but what I mean to say is that suppose I have a E. coli. You know E. coli? You must be knowing the E. coli is a well-known microorganism. Now, suppose I say E. coli grows very faster in presence of glucose. So what it will do is that normally, if I give glucose and galactose in the medium, what it will do is that it will utilize glucose first, and then it will metabolize glucose, produce energy, do its job. On the other hand, if I put lactose, at that time, the enzyme which metabolizes lactose or galactose is not present. What it the enzyme which is responsible for lactose metabolism or galactose metabolism, they will be synthesized, and this way they can control the uh, through transcription process. Okay, now let us move on to another thing. Suppose we need a very quick control. We are not going to wait for the enzyme synthesis. Under those conditions, what enzyme should do? Enzyme can do regulation by allosteric control. So, can anyone one tell me what is allosteric? You have learned this when we were talking about enzymes. So there are a set of enzymes which follow the Michaelis-Menten the kinetics, but there are other which does not follow Michaelis-Menten kinetics. They are allosteric enzyme. These allosteric enzymes will have another site which is known as allosteric site, where either substrate can bind, or inhibitor can bind, or activator can bind. So you have a set of enzyme which is known as, or a type of enzyme which is known as allosteric enzyme. They can control the reaction. So let us take an example how this can happen. Suppose I have an enzyme which can, which is allosteric, which is in and is being activated in presence of AMP. What it means? If some enzyme is inhibited in presence of ATP and activated by AMP. So one is inhibiting, another is activating. So if ATP is inhibiting, that means the enzyme should not work when the excess of ATP is present. And when excess of ATP will be present, that means energy is high in the cell. So if high energy is there, then it will require to go for the metabolic reaction. So that is what is the concept. If ATP is inhibiting, that means it wants the so this way I'll elaborate this concept further. What I mean to say is that when the energy production is required, then the trigger will be different. When energy consumption is required, the trigger is going to be different. So either ATP or AMP, they can regulate the reaction. So this is the way where the same enzyme can become active at one point and inactive at another point. The next concept is that, okay, that is one way of controlling, but suppose I have Michaelis-Menten type of enzyme, or even if we have allosteric enzyme, it is not very sensitive to the energy status. What else it could sense? It could be that the end product of a reaction inhibits the first enzyme. So suppose I have an enzyme which is having several steps. Let me just try to explain it to you. Suppose I have an enzyme which is catalyzing a reaction from A to, say, okay, you have a reaction, and in this reaction you have several steps, and these steps are involving where you have A as a substrate, A is the substrate, and you form B, you form C, you form D, and at the end, your E is the one, or F is the one, which is the end product of the reaction. So what will happen that this, when it accumulates, it is going to inhibit the enzyme which is in the first step. Mostly it is the first step, or alternatively it could be, if there is a branching in the reaction, it will be at the branch point. So that is the way inhibition can occur, which is known as a feedback inhibition. Now, how these inhibition can occur, you will be learning more when you talk about the regulation in the enzyme. That itself is a complete, discipline where we talk about how the regulation occurs. When I'm talking about these, each one could be a full class. So, or maybe more than one class, where we can define how the regulation occurs. But the basic principle is that they can occur at the gene level, they can occur at the enzyme level. If it is occurring at the enzyme level, it could be the energy status which can control, or it could be the accumulation of the product which can accumulate. So there are different ways in which it can be regulated. One of the ways in which enzyme can regulate is covalent modification. So can you recall any covalent modification which we have talked about? When we were talking about the protein, we have talked about that the protein function can change if there is a modification. And one of the modification could be phosphorylation. Do you recall anywhere where phosphorylation was modulating the enzymatic activity? Okay. We have, I don't know whether we have discussed in detail or not, but whenever we talk about sodium potassium transport, the sodium ion is transported across the membrane once sodium potassium ATPase gets phosphorylated. So it is one form of the enzyme. Another form is where phosphorylation is removed from the sodium potassium ATPase. At that time, potassium binds. So what I'm trying to say here is that there could be covalent modification, could be phosphorylation, could be, sometimes it could be methylation, sometimes it could be, but most popular is a phosphorylation reaction. In some cases, there are adenylation reaction. So this way you can control the enzyme. So if I phosphorylate, enzyme becomes active. If I dephosphorylate, enzyme becomes inactive. So this activation and inactivation enzyme plays a key role. Some of these reactions, when we talk about the metabolic reaction, we will elaborate and we'll give you some of these concepts. Now, some of you must be thinking that how enzyme gets a signal that phosphorylate. So mechanism where hormones, they coordinate the metabolic relation between different tissues. So suppose in one tissue you have to change the enzymatic property. So you see the enzyme has already been synthesized, it is already present in the cell, but now it has to be regulated. It has to be regulated in one cell but not in other cell, in one tissue but not in other tissue. So what will happen, that this can be done by modifying the enzyme which is already synthesized. So it is at the enzyme level when it has been synthesized. And most of the time hormones, they do this job. We will cover this in our lectures, that how hormones can regulate the enzyme. That's very critical, because we are knowing that okay, this hormone regulates, this hormone does this function, but actually it is not hormone does the job through enzymes. So they modify the enzyme, and by that doing that, they can control the reaction, or they can control the metabolic pathway. Now, next way could be where the control can occur by regulating the flux of the substrate. Okay, how this can happen? As I told you that if the cell is growing in presence of, say, glucose, and if I say uh, and there is another option that it can grow in presence of galactose or lactose. Whenever they are growing in the presence of lactose, then only they will synthesize enzyme which can degrade them, or which can move them from outside the cell to inside the cell. On the other hand, as long as glucose is present, the enzyme itself is not synthesized. So what has happened? It has a controlled at the level of entry inside the cell. Let us take another example. And that is very clear, and you will find it's a very interesting. The metabolic pathway which is occurring inside the cell, they are compartmentalized. What does it mean? That suppose uh, you have certain reaction happening in cytosol and certain reaction which is happening in the mitochondria. What happens in the mitochondria? Most of the reaction which are responsible for the energy generation, they are happening inside the mitochondria. So suppose I have a cell. Inside the cell, what I do is that I have this as a mitochondria where I will have reaction, which is, for energy generation. And here, outside this cytosol, you have the reaction, which is utilizing the energy. So what will happen? You have compartmentalized. So unless energy, excuse me, unless ATP, which is synthesized inside the mitochondria, comes out, it will not carry out the biosynthetic process. On the same way, unless ADP, which has been produced outside the mitochondria, unless it enters inside the cell, it is not going to be, doing those reaction and producing energy. So there is a tight control in production and utilization of energy. And that is what is the reason why in the higher organism you have a tighter control within the cell. It is not in the tissue, it is in the cell, where certain process is happening inside the mitochondria and certain process which are happening in the cytosol, they are separated. Okay, now my question is, suppose cell has a design where both energy generating process and energy consuming process both are occurring in cytosol. What should happen? Can you think of any, uh, any problem or any solution out of it? If both energy generating process and energy consuming process, they are occurring in the same compartment, what will happen? The energy is being produced, energy is being utilized. So this will be a futile cycle. Can you hear me?
Yes, sir.
Yes, sir.
Okay. So what I'm saying is that suppose ATP is produced here outside the cytosol, and here it is consumed. So in one step it is being produced, in the same step it is being utilized. Rather, here what happens, when there is energy need, only then ATP will come out. There is a very strict mechanism by which ATP, ADP translocation occurs out inside and outside the mitochondria. But that's a mechanism by which it can control the reaction. So you have learned the concept why the compartmentalization occurs in the cell, why the energy generation process and energy utilization process has been designed to happen in different, compartments. Same way, you will be seeing that the tissue which is carrying out the synthetic process will not be mostly utilizing the energy. They will be synthesizing macromolecule. On the other hand, some of the, tissues, they will be responsible for energy metabolism. Now, how the cell gets or sensed whether the energy is being produced or energy is being utilized? What could be the actual way by which you can see that energy status is high or low? So here there is a certain index which is given. There are two different types of index has been given. One of them, which is commonly used, and which you can see whether the energy status of the cell is high or low, is based on energy charge. And energy charge is taken as a ATP plus half ADP concentration divided by the concentration of ATP, ADP, and AMP. So if you look at, if I have all the molecule as a ATP, that means it is high charge. If all of them as AMP, that is a very low charge. So this charge, this value can be assigned from 0 to 1. So all AMP means 0 energy charge. If all are ATP, that's the 1 charge. So what should we expect? What should be the energy charge of the cell? Let me tell you that the energy charge is being regulated. So whenever you have high energy present, too much of ATP is present, they will go for uh, they will when too much of energy is present, they will go for ATP utilization, and whenever there is a too less of ATP or all AMP is present, then they will go for ATP synthesis. So it's very clearly shown that ATP generating or catabolic processes are inhibited by energy charge. So that means uh, the when energy charge is more, it will stop the catabolic pathway, that means ATP generating process. Rather, they will go for anabolism, that means the one which is synthesizing macromolecule. Whereas ATP utilization or anabolic pathway are stimulated by high energy charge. So one is inhibited, another is activated. So how it works? It works like a bulb. So in the cell, energy is that energy charge is mostly maintained between 0.80 to 0.95. So that means they are neither having one nor having zero. They are in between. There is another index which has been used, which is known as a phosphoryl potential. So, or phosphorylation potential. Now, this phosphorylation potential is given by the just by considering ADP and ATP, and their so when ATP is hydrolyzed, what will happen? They will produce ADP and PI, and on the other hand, you can have all ATP if it is not being utilized. So this is another index, and that can be very easily monitored inside the cell. So that is why it has been, given as an index for considering how the metabolic processes is happening or what is the energy status of the cell. So now with this, I have given you the basic concept of metabolism. It gives you what is the introduction, how the metabolism occurs inside the cell. After completing this, because we don't have too many classes and even otherwise, it is important to consider the concept that how we study enzymes, because enzymes are going to play a major role in metabolism. So we need to understand how enzyme works. And to understand the enzyme, how it works, you have learned some of the kinetics, some of the information, but you do not know how we can study enzyme. So suppose I take a cell, there are hundreds and thousands of enzymes. So how to study those enzymes? Can we purify? Can we isolate a single enzyme and study them? So that question is answered by understanding how proteins or enzymes are being purified. So our next talk is going to be on how enzymes and proteins are being purified. So we will move on to that. If you have any question, I would like to address.
Sir, is this energy charge is just an index, or does it have a physical significance?
So it is an index, and it has also a physical significance that a cell, if it is not metabolizing properly, you can understand that, okay, cell is not, metabolizing properly. It needs something. So suppose its energy status of the cell is very poor, you can get to know. So this value, which is 0.80 to 0.95, it is given to tell you about the phosphoryl group transfer potential. But at the same time, it is an index. It can tell you under what condition the energy charge cell is having full energy charge, or under what condition cell may not have energy charge. But actually, all the ATP present, this is the value. But ultimately, in a normal functional cell, it is going to be controlled. It is going to be buffered. Anything else? You have learned the concept, if that concept of regulation is clear to you, because that makes a big difference when you will study enzyme, metabolic pathway or metabolic engineering. So this is a very basic concept which tells you that the regulation can occur at different levels and how that level they are going to perform. I have not told you how the transcriptional regulation occurs. That you will study when you study your genetics and molecular biology. Then same way, I have not told you that how the the last step of the any enzymatic pathway is going to control the first enzyme. And if you look at, there is a certain significance of why the last one is inhibiting the metabolic pathway. Why not that A to B is formed and B inhibits the first enzyme? If you look at the logic is that if there is a metabolic pathway, suppose I'm talking about glucose metabolism, there are 10 steps involved in that metabolism. And what I'm doing is that if I'm inhibiting first step, even after the second step is formed, what will happen if that there is a branching in the pathway, the branched molecule, they will not enter in another pathway. So this will be inhibiting all the reaction. Second thing is that you may argue that, okay, let us inhibit the last step in the pathway. That will happen. If that happens, what will happen is that it will be synthesizing all the enzymes, all the enzymes in the pathway. That means more energy will be required for synthesizing those enzymes. So actually what is happening is that this mechanism of control by the last by the pathway to the first, basically control on the energy consumption. That means you are protecting, or you are saving all the enzymes which otherwise would have been utilized. Is it clear to you? I don't think that it is clear to
Yes,
you.
sir.
What I'm saying is that if I have a step A to B and I have enzyme E1, then I have a step B to C, I have enzyme E2. Step 3 is C to D, you have enzyme E3. Now, in the last step, if D is inhibiting, for example, E3, what will happen? E1 and E2 is already synthesized and there will be waste. So what it does is that D will inhibit E1 so that E2 and E3, they can be saved. Okay. Okay, let's move on to the next chapter. We have almost half an hour, so let us move on to another very interesting uh, thing which is important. And most of the biochemists, or in those who are learning biochemistry, they some way or the other they are involved in protein separation and purification. You will also do a couple of experiment on protein separation and purification, at least separation part in your experiments. So let us talk about what is the source of protein and enzymes. So if I talk about enzymes, I'm talking about all the enzymes which are proteinaceous in nature, because 99% enzymes, they are proteinaceous in nature. So we are considering, when we are talking about enzyme purification, when we are talking about metabolic regulation, we are talking about the purification of protein and enzyme in the same way. The only difference between protein and enzyme separation at the end will be that when you are separating protein, it is relatively difficult to monitor their activity. When I say activity, means that what is the functionality of the protein. So you have a complicated test. But if I'm purifying enzyme, it is very easy, because out of that fraction, I can say that this is the enzymatic activity. And how do we know about the enzyme activity? You have done the we have covered these enzymes. How we get to know the activity of enzyme? I think you have not revised what you have learned. So whenever we talk about the activity, we look at the, you have a substrate and your product is formed. So what we do is that either we measure the formation of a product, or we measure the consumption of the substrate, by which we can get to know what is the activity of enzyme. And activity you have learned by, how we can find out? We can find out by learning about the velocity of the enzyme catalyzed reaction. So we have talked about Km, we have talked about the velocity. So in enzyme catalyzed reaction, if I say that this is the velocity, that gives you, terms in terms of what is the activity. So it will give you that how much product has formed per milligram enzyme. I'm you the how calculate activity of enzyme. These things we will discuss more when we go for purification, but you should know the concept, basic concept. So where proteins are present? Proteins are present mostly in tissues or in microbial cell. When I talk about tissue, I mean both plant cell as well as microbial cell. And when I say microbe, I do include everything. That means fungi, bacteria, everything. So any microbial word we are talking about, any plant tissue or any, any animal tissue. So all of them, they will have protein. So that is the source of protein. So if I want to purify protein, or for that matter enzyme, the first step in any protein purification procedure is to break open the cells and release their protein content in a solution which we call as a crude extract. Suppose you are growing E. coli or any other bacteria, say bacillus, what you do is that you grow them in a flask. After 24 hours or 4 hours or whatever, you know which cycle you are collecting the cells, and then you break it open. And if you break it open, you will have hundreds and thousands of enzymes and proteins present in that solution. Now the question is how we will get to isolate one particular enzyme or one particular protein which is of our interest. It's a very tricky process. You are trying to isolate one of the, the component which is of your use. Some of you may ask, why should be interested in isolating a protein or an enzyme? Answer is very simple. If I want to learn about the protein, if I want to know which protein is controlling the activity, if I want to know which protein is getting activated or inhibited, we need to purify them. We need to test them and see how they are behaving when they are present as an individual. Otherwise, there are set of enzymes there. So suppose glucose is forming pyruvate. How will I get to know that there is only one, there are these many enzymes there involved? Why not that there is a single enzyme which is converting glucose to the pyruvate? So what we do is that we purify all the enzymes. But at the same time, if I have to determine the metabolic pathway, can anybody tell me that if I have a reaction which is going on inside the cell where A is converted to B, B is C, C to D, D to E, is there any way to monitor that there are four-step process or a single step process? Any one of you? What could if I get inhibitors for all the reactions separate? Suppose step A is inhibited by X, step B is inhibited by Y, step 3 is inhibited by Z. We know that there are three different enzymes. Other way could be that you can create a mutation. That's another way of finding out the metabolic pathway, where you find out the mutants. And mutants, why mutants are formed? Because you have inhibited the gene, or your gene is not synthesizing that particular enzyme. So there is a mutation, and because of the mutation, you get those mutants which are defective in the pathway. So what they will do, if I get mutant of the first step, that means they are not metabolizing the reaction. If I get mutant of the second reaction, so what they will do is that they will synthesize the product of the first reaction. So first reaction has occurred, but the second reaction onwards has not occurred. Now I get another mutant. So what they will tell you that the third step has been blocked and was not present. This has been done. And another would be we have a radioactive level where we can, individually trace them, and that's known as a tracer technique. So there are three ways by which you can get to know that yes, actually metabolic reaction involves many, many steps. So what you do is that you make a crude extract. So suppose I grow E. coli, I will take out, I will burst the cell. Bursting again requires different techniques. It could depend on how hard or how soft your surface of the bacteria is. Suppose I have to break a bacteria, I may use sonication. Suppose I have yeast cell where I have very hard, cover, then I may use another technique. So there are many techniques which are used for making the crude extract, but most common one is the sonication. Now, once you have got the crude extract, next step is that you separate the protein into different fractions. And these fractions will be based on the property of a protein. Could be, we can separate the protein based on their size. We can also separate protein based on their charge. And whole process by which we are separating these fraction is known as a fractionation. An early fractionation step in the purification. I'll tell you that there are different steps in protein purification, and there may be different techniques or different combination of techniques which can be utilized for purification of a protein. An early fractionation step in purification utilizes difference in protein solubility. So if I have two proteins or 100 proteins, out of which one is less soluble, I will solubilize all other protein, and the one which is not solubilized will be taken out. So this is a complex function of pH, temperature, salt concentration, and some other factors. So here, a very interesting concept which you have learned in your chemistry is used, which is known as a salting out. Do you remember what is a salting out? Any one of you? It is written in your PowerPoint. You can see the solubility of a protein is lowered in the presence of some salt. It is called as a salting out. So the question comes, why the solubility of a protein is lowered in the presence of some salt? You may have also learned about salting in. That phenomenon is because of the more protein solubilization when there is some salt in a certain concentration. But most common phenomenon is a salting out, where you put the salt and protein gets separated or precipitated. Can you tell me why should it happen? The addition of certain salts in the right amount can selectively precipitate some proteins, whereas others remain in solution. And most common salt used is ammonium sulfate. Anyone? Now, I would like to know from you a very basic concept. If protein is a soluble in water, why it is soluble?
So because the interactive sides are facing outwards and the hydrophobic sides are like facing inwards, called inwards.
No, this is the structure. Why it is soluble?
So because they can interact with the water molecules, right? And like
Okay.
hydrogen bond with them.
Okay, that's fine. So the same way, whether you think of sodium chloride, why it is soluble, or you think about a protein, the same basic concept applies. So if everything is present in the hydration cell, so every molecule will have a hydration cell. And if lot of water is a binding to the surface, they will be the one which will be soluble. So any protein which is having lot of binding with the water, they will be solubilized. Now, I want to make them insoluble. So what I should do? I should remove those water molecules which are binding with the surface of the protein selectively, and that will allow that protein to precipitate. Am I clear? So what salt can do is that salt will compete with the protein in binding with the water. So if X amount of water is a binding to the surface of a protein, they will be detached, they will be binding with the salt, and that is the where, because the surface of the protein has been emptied from water. So what they will do? They will salt out, they will precipitate. So it is actually competition between the protein and the salt towards the water which is present on the surface, and that is where the salting out phenomenon occurs. Now you may say, why salt should replace water from the protein? The answer is very simple, that protein is a bigger molecule, salts are a small molecule, so they will be, their number of the salt will be much larger than the number of protein. So if one protein is there, you can have several salts which will be competing, and that's where they can replace the water which is present on the surface of the protein, and that's the way they make them to salt out. Now, there could be another way of purifying protein from the smaller molecule. So suppose you have done the salting out. Now this salt may interfere in the later step in the purification. So what we do is that we separate the smaller molecules from the larger molecule. Proteins are always larger than the salt. So what you do is that you carry out a process which is known as a dialysis. So it's a very common process. You will get the dialysis bag, and what you will do is that you will put your protein solution, put them in a buffer and leave it in the buffer for a longer time. What will happen? All the salt will be moving out, where proteins will be retained in the your bag which is semi-permeable, made up of semi-permeable membrane. So dialysis is a procedure that separates protein from smaller solutes by taking advantage of protein larger size, and the partially purified extract is placed in the bag or a tube made of some semi-permeable membrane. It is available, it's synthetic membrane which is available. And then what you do is that you suspend them in a larger volume of buffered solution of ionic strength membrane proteins. So that's the way you do dialysis. Let me tell you that this time I'm not teaching you how you can separate two proteins by using membrane. So it is also theoretically as well as practically possible to separate two proteins of two sizes by using a membrane. So you have a membrane which has a certain cutoff of the size, and that's the way it can be separated. When you study membrane science and engineering, you will understand the concept in detail, that how proteins can be separated using membrane technology. And that's a very powerful technology because it can have possibility of scaling up. So that process you will learn when you learn downstream processing or membrane based purification. Now we are going to focus on column chromatography, a simple chromatography where you have a column, and then using column chromatography, you take the advantage of protein charge, so you can separate them based on the charge. So by the way, why protein has a charge? Why any protein should have a charge? Proteins are made up of amino acids, and of course, there will be one positive charge and one negative charge at the end of the protein molecule. In the beginning, you have starting with NH2, which becomes NH3. So you have one positive charge and one negative charge. But other than that, why there is a charge? Because that will be net charge will be zero if I consider just ammonia, this N terminal and C terminal. Okay. Here, the charge will be contributed because of the R group of amino acid. It's not amino acid alone, because there are certain amino acid which will not contribute. So suppose R group is not having any charge, like alanine, valine, leucine, etc., they will not contribute towards the charge. But if I have a positive charge protein, amino acid, like histidine, arginine, so they will be, or lysine, they will be contributing to the positive charge. Whereas amino acids like glutamic acid, aspartic acid, they will be contributing towards the negative charge. And then it is a sum of these number of amino acids. So suppose I have a lot of glutamic and aspartic acid in a protein and less number of lysine, histidine, and arginine, what will happen? The charge on the surface of the protein will be negative charge. So each protein will differ in terms of their charge on the surface. And that's where you have a charge in the protein. Size you can very well understand, depending on the number of amino acid, their size will be different. So that is determined by the kilodalton, how many kilodalton they have. The third one could be binding affinity. Tell me why protein should have any binding affinity. Can you tell me any protein which has a binding affinity? How can we separate based on the binding affinity, and why should they have binding affinity? You have already learned about lectin. Do you recall? We have talked about something which can bind with the glycoprotein. So they are lectins. We have also talked about that the enzymes, they have a specificity to bind with the substrate. So which, if I create a molecule which has a structural similarity with the substrate, they can bind with the enzyme. So these are the properties by which the enzymes can bind with the, your enzyme which you are interested in separation can be allowed to have certain affinity. If there is no affinity, there is a way by which you can introduce affinity in the protein. So what is done is that most of the time you purify the enzyme, and if it is a recombinant process, which you will be learning how we can do that. So you take a gene, and in this gene, why insert something where we can put a number of histidine molecule, at least 5 histidine is introduced. And if I introduce it, histidine has a specific affinity for nickel. So what will happen that if I have a protein where histidine tag is present, we can separate them based on their affinity for histidine. So we make a column which is made up of nickel NTA, and they can bind with the histidine part of the protein, and then protein can be separated. So these concepts we will be elaborating in the next few lectures, and today, I'm just introducing that the column chromatography is a powerful tool for purifying enzymes. And so they are based on their property, you can separate them. So what you do is that you make a column where you fill with the some kind of solid material, which is a stationary phase. And then from the top of the column, you pour the buffer, which is called as a mobile phase. And once mobile phase migrates, and in this mobile phase, if I am having proteins present and it is a mixture of protein, so they can be separated because mobile phase you can collect at the end, and individual protein, depending on their migration, you can separate this out. So this PowerPoint gives you an idea. We will be elaborating on this further. But think of a scenario where I'm giving you a column, which is the picture has been taken at 5 different stages, and each one is explaining that how the proteins are going to be separated. In the first one, I have put the protein in the column at the top. So column is made up of a glass or plastic. Inside, we have put the gel material, and on the top, we have put the protein mixture. And then what we are doing is that from reservoir, we are pumping buffer, and by pumping buffer, you have seen in the first one, there is no separation, just you have matrix. In the second one, you have seen some separation. Third one, separation has become more. Fourth one, it is a further. And fifth one, you have got one of them separated out. So this is the process, and how you can do that, how you can monitor whether it has been separated or not. You send it to the detector, you have a column, fraction collector, etc. So I will talk about this concept further. So I'm stopping at this point. If you have any question, please let me know. Any question or any doubt? So if you do not have any question or any doubt, then I will sign it off, and we'll meet. I think tomorrow we have a class, so we'll meet in tomorrow's class.
Thank you, sir.
Thank you. Thank you.
Thank you,
I'll
sir.
sign off now. Okay. Bye.